Hi, today I'll be illustrating the synthesis and cellular movement of some key macromolecules. The first is a lipid, cholesterol. The second, a secretory protein, insulin. And the last, a type of intracellular protein, ribosomal proteins. The accompanying flowcharts will help outline important steps and enzymes in the synthesis pathways. Cholesterol is an important lipid for its role in plasma membrane stability and as a precursor for steroids, vitamin D, and bile acids. While all cells do contain enzymes for cholesterol production, since it is a vital component of the plasma membrane, it is most actively produced by those cells in the liver, adrenal glands, intestines, and gonads, which require production of various steroids. Cholesterol biosynthesis begins with acetyl-CoA, which is produced in the mitochondria through the oxidation of fatty acids, pyruvate, or amino acids. This is then converted in the endoplasmic reticulum to acetyl-CoA by the enzyme acetyl-CoA acetyltransferase 2, or ACAT2. And this is the starting point of a series of reactions that becomes part of the mevalonate pathway, leading to the production of cholesterol. 3-hydroxy-3-methylglutaryl, or HMG-CoA synthase, converts acetoacetyl-CoA to HMG-CoA, which is then acted on by HMG-CoA reductase to make mevalonic acid. HMG-CoA reductase activity is inhibited by glucagon and activated conversely by insulin. Its synthesis is also regulated by a transcription factor called steroid response element binding protein, SREBP, which you can see in step 8A. Mevalonic acid continues through the pathway to be converted through several more enzyme-mediated reaction steps into cholesterol. So we see energy being used to convert mevalonic to 6-isopentenyl pyrophosphate, and then that's converted to squalene and then lanosterol, which is finally, I think, after 18 plus or more enzymes in a pathway, lanosterol is converted to cholesterol. And then cholesterol production itself, as you can see in step 8A, imposes negative feedback on its own pathway by binding to a steroid binding protein called SCAP. This is needed to activate SREBP, which we discussed earlier, to produce HMG-CoA reductase. So when SCAP is bound to cholesterol, it is not free to activate SREBP, and the pathway is essentially shut down right before step 3. Cholesterol is transported from the endoplasmic reticulum via vesicles through the endomembrane system or by lipid transport proteins, LTPs. Some of these are part of the Gram-D1 family, which move cholesterol between the endoplasmic reticulum and the plasma membrane. And that is noted in step 8B. And then within the cell, free cholesterol is not stored as cholesterol, it's stored as cholesterol esters, which need to be acted on cholesterol esterase to be converted to cholesterol free. And that is in step 8C. Next, insulin is a protein that is essential to blood glucose and metabolic regulation. And the gene coding it is called INS. It's on chromosome 11. Insulin is produced and secreted by pancreatic beta cells. First, the amino acid sequence for preproinsulin, a precursor that's composed of a signal peptide, in insulin B chain, a C peptide, and an insulin A chain, is translated from mRNA by a ribosome on either the rough endoplasmic reticulum or less often in the cytosol. Protein chains produced in the cytosol are guided to the endoplasmic reticulum by, or ER, by their signal peptides, which are recognized by regions on the ER called translocons. All of the preproinsulin, whether it's produced on the ribosomes in the endoplasmic reticulum or in the cytosol, all arrive at these translocons. Specifically, the regions are SEC61 and SEC62. Translocation proteins that include some names such as HSP70, TRC40, calmodulin, and TRAP are some that facilitate the movement and docking of preproinsulin to the ER membrane. Within the ER membrane, preproinsulin is cleaved to form proinsulin. Proinsulin then undergoes a series of folding activities. So this is assisted by the polymerase disulfide isomerase family of oxidoreductases, and then it's also assisted by other proteins in the endoplasmic reticulum. Several of these also monitor the folding for errors such as BIP and P58IPK, and they do so by binding or assisting the binding of other proteins to hydrophobic patches on misfolded proinsulin. Proinsulin then enters the cellular secretory pathway when it's transported in a vesicle to the Golgi apparatus. It's then sorted and packaged into immature secretory vesicles. Within these vesicles, enzymes such as prohormone convertases, such as PC1 and PC2, and carboxypeptidase cleave the C peptide from proinsulin. So this results in the B chain and the A chain still being intact, and that's what we call insulin. Insulin remains in storage then in these vesicles, these mature vesicles, until it's signaled for secretion. And how that happens is as we consume food, our blood glucose rises, and some of that glucose is transported into the beta cells through the GLUT2 transporters. As glucose metabolism increases free ATP in the cell, this signals some of the leaky potassium channels to stop pushing potassium out of the cell, so they're inhibited, and also triggers the opening of calcium channels, which allows an influx of calcium ions into the beta cell. The influx of calcium triggers the docking of insulin storage vesicles to the cell plasma membrane so that they can finally merge with the plasma membrane and release this insulin through exocytosis. And lastly, the intracellular protein that I took a look at was a type of protein called R proteins. I feel like in many biology courses, rRNA and the structure and function of ribosomes is sometimes only vaguely approached, but ribosomes are so important to all cell operations that require any protein synthesis, so I wanted to touch upon this a little bit. Ribosomes in the eukaryote begin their assembly within the nucleolus, which is a region located in the membrane-bound nucleus. The region itself is not membrane-bound. It's just the area that contains DNA that codes for ribosomal RNA or rRNA. Ribosomes are usually made as two distinct subunits, a small 40S subunit and a large 60S subunit using rRNA and ribosomal R proteins. The subunits then combine with mRNAs in the cytosol to form functional ribosomes. And they only come together when there is mRNA present that needs to be translated. The 40S subunit is made up of 47 distinct R proteins, while the 60S subunit consists of 33 of those proteins. The majority of R proteins are produced in the cytoplasm by translation of mRNAs transcribed in the nucleus by RNA polymerase II. mRNA that codes for R proteins engages with existing 40S ribosomal subunits in the cytoplasm, and then they recruit a 60S subunit. And then with the assistance of tRNA, the mRNA is translated into amino acid sequence, which is finally released and then folded and transported back to the nucleolus for assembly of ribosomal subunits. And then R protein translation is regulated by some initiation factors. They fall in the family of 4EBP proteins, and they bind to the M7G cap, which is common to many mRNAs, and specifically to R protein mRNAs. And they help recruit RNA helicase to untwist the mRNA and make it more easily accessible. The sensitivity of R protein mRNAs is cap dependent, and this regulation, according to Nee and Bushak, creates a feed-forward loop. So increased availability of ribosomes and translation initiation factors strongly promotes the translation of R protein gene mRNA. So the more ribosomal ingredients you have, the more you have an ability to actually translate our proteins and then create more ribosomes.